Hi everyone, I am Ikoyo So Philip of Team 6 in the affirmative group, and with me is Peter Esama. Together, we are going to be presenting on the topic: should genetically modified microorganisms be widely used to combat antimicrobial resistance? So, briefly before I start, I would like to give us a brief introduction of what this is all about. So, what is antibiotics resistance? We all know that most times when we fall ill, our first line of thought is to get ourselves antibiotics to get treated for infection. So, antibiotics resistance has now become one of the most pressing global health challenges, which occurs as a result of the overuse and misuse of antibiotics in clinical settings, agriculture, and animal husbandry. And this has accelerated the emergence of resistant bacterial strains. Currently, traditional antibiotic development struggles to keep pace with evolving resistant mechanisms because as often as we expose ourselves to these antibiotics, the bacteria mutate and they become more resistant to the antibiotics. And so our conventional medications are struggling to keep pace with this. So genetic engineers are rising to this challenge by using innovative approaches, leveraging on genetically modified microorganisms. So genetically modified microorganisms are basically microorganisms modified genetically through gene editing or gene knockout or knocking just to enable us combat antibiotics resistance. So, genetically modified organisms are engineered to tackle antibiotics resistance by either enhancing therapeutic efficacy or neutralizing resistant pathogens. Of course, we do have a growing global concern, crisis that requires novel intervention. So, we cannot just remain on the small scale, traditional small molecule antibiotics to address the situation. So, we are therefore proposing that GMMs, which is genetically modified microorganisms. Subsequently, I'm going to be calling them GMMS, which have been strategically designed with a proven efficacy in both laboratory and field settings, should be used to combat antimicrobial resistance and reduce mortality resulting from this situation. So we are going to be arguing these points based on the fact that we know that a conventional um, medicine are no longer able to meet up with the evolving pace. So our first point of argument is precision targeting. So engineered bacteriophages and CRISPR-based interventions have allowed for high specificity in eliminating resistant bacteria without harming beneficial microbes. Unlike the broad spectrum antibiotics, which disrupt entire microbial ecosystems, of course, we have some of our medications that when we take them, we find out that we have some other resulting symptoms, which is as a result of the antibiotics that we took. So it was aimed for one thing, and then it has disrupted the microbial ecosystem. But with GMM, we would have them specific, targeted at the particular mutated bacteria which is responsible for antibiotic resistance. And it has also been recorded that GMM in the gut have been shown to preserve gut microbiota and and environmental balance. And some of these GMM are CRISPR modified bacteria for gene editing. Of course, I expect that the opposing team would want to argue that some of the World Health Organization therapy are phages therapy, and CRISPR edited bacteria. Let's not forget that CRISPR edited bacteria are also genetically modified bacteria because CRISPR itself is a genetic tool that is used for gene editing. So anything that has had his gene modified or edited is actually a genetically modified organism. The next one is also for overcoming resistance. We know that traditional antibiotics often become obsolete as bacteria mutate because as we keep exposing ourselves to the antibiotics, bacteria mutate and this becomes obsolete. So GMMs will provide adaptive solutions that can restore antibiotic sensitivity by editing bacterial genomes or disrupting resistant genes. This keeps the antimicrobial intervention effective in the long run. Example of such has been engineered bacteriophages that target and kill antibiotic resistant bacteria, and there have been reports of them being done where engineered bacteriophages were targeted at specific sequences of the mutated bacteria, the resistant bacteria, and not other bacteria within the ecosystem, and they have recorded positive results. Another constructive argument we would be putting forward is reducing antibiotic dependence. The overuse of antibiotics in healthcare, agriculture, and livestock is the driving force behind resistance. But with GMM-based therapeutics, it will reduce reliance on antibiotics because this is not just like a medication that you just go over the counter to take. No, you have to be recommended for it for you to be able to have access to it. So it's going to reduce reliance on antibiotics, leading to fewer cases of antibiotic resistance in the long run. We also argue for eco-friendly and sustainable therapeutics. Of course, we know the cost, the chemical production involved in our conventional antibiotic production, which is not environmentally friendly. It's arguably environmentally taxing. But with GMM based solutions, we are going to be leveraging on biological factories. So that means it is done in the factory that generates antimicrobial compounds in a sustainable manner with minimal waste or pollution. So, of course, who wouldn't opt for an eco-friendly and sustainable alternative when everybody's all about climate change, climate preservation, and the rest? So this offers us an opportunity to do something that is also eco-friendly. We're also arguing that it's going to be combating biofilms. We know that one of the major causes of um, bacterial resistance is when they produce biofilms. So these mutated bacteria, when they are first exposed to antibiotics, because they've mutated, they produce biofilms that coat them and protect them from the effects of the antibiotics. But with engineered microorganisms, we can dismantle these biofilms, exposing the bacteria to treatment and preventing persistent infections. And by so doing, it reduces the the exposure to antibiotics, which over time causes antibiotic resistance. So, of course, we have a couple of rebuttals that we want to address. We are not saying that GMMs are the best thing after sliced bread. Of course, no. We know that for everything there is a pros and a cons. And these are a few points that we're going to be using to present our case as per the rebuttal that has been put forward. We have the rebuttal of biosafety risk. We are arguing that with this biosafety risk, we can use control contaminants and precise engineering to address this. We know that there is fear of unintended consequences of releasing engineered microbes into the atmosphere or into the ecosystem, but with rigorous biocontainment protocols can mitigate this risk. Another thing is that GMMs are designed to be self-limiting in that they are programmed for cell death and they are dependent on certain synthetic nutrients which are not found in the natural environment. So if GMMs are out there in the natural environment, they will not be able to survive. So there's going to be programmed self-death. And then also, CRISPR-based editing can be highly precise, minimizing off-target effects. Of course, we argue about unintended consequences, but with precision, we can minimize this off-target effect. And there are some of this that are already there facing clinical trials, which is World Health Organization is already considering them because they are precise in terms of targets of the mutated bacteria. We also have the rebuttal of regulatory gaps and biosecurity. There are already existing frameworks and advancing policies concerning GMOs, concerning biotechnology as a whole. So when we argue about this, let's not forget that we understand that regulation is crucial. We are not just saying that anybody can go into in his or her lab, make a GMM and bring it out there for us. No, we know that regulation is crucial. So that is why we will leverage on existing extensive protocols for GMO safety, even with these GMMs. GMMs is not an exception. So existing protocols would also be governing GMMs and need to continue to evolve to accommodate issues arising. The same rigorous testing standards that are applied to vaccines, that apply to therapeutics, would also apply them to GMMs to ensure safety before it's being exposed to human or livestock applications. So we are not just saying because it's GMM, get out there. No, the same existing protocol, the same rigorous testing that involves them like clinical trials, phase 1, phase 2. GMMs would also be subjected to this until proven that they are safe and can be used before it can be applied in clinical settings or in agricultural settings. We can also address ethical concerns through transparent research practices. Of course, if we have a transparent research practices, if we have bodies governing this and ensuring that everybody's doing a transparent research practices, this can also be addressed. So, I mean, the issue of regulatory gaps and biosecurity, we can address this with existing frameworks and advancing policies, which is why we are all here. This is why the essence of taking this course, because we are advocating for these policies to be implemented. And we know that these policies will not only safeguard the experiment that we do, but will also ensure that the public get the maximum benefit of what we are producing. The next rebuttal was uncertain environmental impact and horizontal gene transfer. Of course, there is reduced broad spectrum disruption. As much as we say that there'll be environmental impact and horizontal gene transfer, let's not forget that traditional antibiotics also indiscriminately kill bacteria. Yes, leading to dysbiosis in human microbiomes and environmental contamination. But with GMMs, we can always address this. With traditional antibiotics, this cannot be addressed. But with GMMs, we can address this, which will allow for targeted interventions, minimizing ecosystem disruptions. Before laboratory findings are introduced to the public, they would undergo thorough field and clinical trials to ensure reduced negative ecological impacts. And additionally, engineered microbes can biodegrade faster than persistent antibiotic residues, reducing long-term environmental effects. So the reduced broad spectrum disruption can also be of help. And then ethical concerns and public trust. Public concerns and public concerns about GMOs often stem from misinformation. Where there is doubt, there will be consent, there will be distrust. But with education and proven benefits, we can be able to better advise the public. We can address this by going back historically, looking at the advancement that has come from GMOs, such as insulin. We have insulin producing bacteria, and that has been successfully used in clinical settings. Even the latex that we use for suturing surgery is also genetically produced. So these have been going on for a long time. So with robust educational campaign along with real world applications, it can prove the efficacy of GMOs and reducing resistant infections. And with this, the society can come to accept it and it can shift perceptions positively. Antibiotic stewardship. We argue that this can also, with this, we rebut this that scalability, that we, it's against antibiotic stewardship, we can look at scalability and lower long-term costs. We all know that prudent use of existing drugs over time has led us to this issue. The so-called prudent use of existing antibiotics has led us to this issue of antibiotic resistance. Of course, investment in new antibiotics with better therapeutic advantage over the traditional ones is the goal. And GMMs are definitely in line with the current need. The initial development may be costly, but biological manufacturing is more sustainable than chemical antibiotic production, reducing supply chain dependency. With advances in synthetic biology, automation, engineering microbial solutions can be mass produced affordably, making them accessible even in low resource settings. So, of course, antibiotic stewardship, we can have scalability and lower long-term costs. So, in our closing statement, while we understand that there are concerns about GMMs and these are valid, there are also current and emerging innovations which are effectively addressing this risk. We are not saying that GMMs are out there, so we should use it, just any GMMs. No. With proper oversight, containment strategies, and public engagement, GMMs have the potential to revolutionize antibiotic resistance management in safe and sustainable manner. So yes, antibiotic resistance is a real-time problem facing us globally. At the same time, GMMs are here to stay. And so what we can do is to make sure through thorough regulations and policies, we make sure that we get the best out of this engineered microorganism to be able to combat antibiotic resistance. Thank you.